Alright, welcome to a COMLEX review of the cerebellum and basal ganglia for the second year students getting in for COMLEX. Hope to do this in person, but as you guys know, things have kind of gone sideways. So let's do our best to kind of keep plowing through and address the crisis in the best way I know how, by teaching anatomy. It's not the best skill set, but it's the one I got. So let's talk cerebellum to start with. Largely, we have the cerebellum to thank for some motor planning, but largely the editing and improvement of ongoing motor activity, whereas the basal ganglia is involved with a lot of things, but we're going to focus mostly on its role in planning and initiating movements, and the movements themselves are then going to be triggered by the frontal lobe, particularly the primary motor cortex at the precentral gyrus. So let's go ahead and see what we've got here. In terms of the anatomy of the cerebellum, it's got very big cerebellar hemispheres shown here, with a lateral part on this side of the dashed line and an intermediate part on this side of the dashed line. And right on the midline, we have an area called the vermis, which means worm. So someone looked at this and thought it looked very wormy, which, all right. In addition to that, there are numerous folia, these little gyri sticking off of it on the surface where we find the gray matter of the cerebellum. Now, there are some other fissures, like the primary fissure here, but we're not going to worry too much about that as we go forward. We're going to revisit the blood supply at the end, but just quickly, it's worth taking a side view of the cerebellum, seeing the lateral hemisphere here, and noting that it's connected to the brainstem by an inferior cerebellar peduncle, connecting the medulla to it, a middle cerebellar peduncle connecting the pons to the cerebellum, and a superior cerebellar peduncle which largely connects it to the midbrain. And nearby we have the vasculature. The vertebral artery gives off the posterior inferior cerebellar artery, the pica. And then generally the basilar artery is going to give off the ICA, anterior inferior cerebellar, and superior cerebellar artery, or SCA, right here. Now, we're going to see a few of the other structures in the cerebellum that are hidden on its anterior or ventral surface. We're going to chop it off right there. And on the brainstem side, we can actually see that superior, inferior, and middle peduncle more clearly. You can also see the fourth ventricle that's shared between the cerebellum posteriorly and the pons and medulla anteriorly. So here's the same view on the other side. We have the vermis on the midline, intermediate part of the lateral hemisphere here, lateral part of the cerebellar hemisphere here, and we have a couple little bumps that are going to come into play a little bit later. On the midline, we have the nodulus, and on the left and right side, we have the flocculus. And altogether, we call this the flocculonodular lobe. It's an important functional part of the cerebellum. And right here, the most inferior part of the cerebellum is going to be on the midline, or near the midline, is going to be the tonsils, which can herniate through the foramen magnum whenever we have increased intracranial pressure. That is one possibility. All right. So let's talk about the actual circuitry that lets the cerebellum do its job. We have two inputs, period, mossy and climbing fibers. So those are the inputs, and the important thing to keep track of is that they operate on kind of an on-off method that we see repeated from pretty much the inputs to the cerebellum in general, as well as within subdivisions of the cerebellum. Essentially, these mossy and climbing fibers are going to activate structures called the deep cerebellar nuclei. Then those same inputs will go out to the cerebellar cortex, which will then eventually cause an inhibition of this deep cerebellar nucleus. So we have kind of an on-off method to modulating activity from the cerebellum. And we'll see that again at a lot of different levels. Pretty much every input to the cerebellum is what's known as a mossy fiber, except for those coming from an area of the medulla called the inferior olivary nucleus, which are called climbing fibers. So once they get into the cerebellum, they behave in similar ways in the gross sense here on this slide, but we'll go into the particulars of how they look different and how they act differently when we get to the actual histology of the area. So the mossy fibers are going to be any inputs not from the inferior olive. So let's start with those coming from the pons. So the pontine nuclei are clusters of cells throughout the tegmentum of the pons, and we have running through the brainstem the corticospinal tract, and those fibers are traveling through the cerebral peduncle, through the pons, through the pyramids of the medulla, decussating at the base of the medulla to get to the spinal cord. Alongside those, we have fibers from the same areas of the cortex that make it to the pons, but at that point they're going to synapse with pontine nuclei. Those pontine nuclei will then decussate across the midline, go to the opposite middle cerebellar peduncle, and then reach to the cerebellum. So those are the pontocerebellar fibers, and they're massive. They make up the huge middle cerebellar peduncle. For the next part, you've got a choice. You can kind of follow along with me on this slide where we're going to visually look at the different tracts that are traveling from the spine up to the cerebellum, or you can jump to the next two slides, which are the verbal bullet-pointed breakdown of these pathways. So do whichever one works best for you, or try a little bit of both. So here we're going to start by looking at the mossy fibers coming from the dorsal spinocerebellar tract. So this tract is originating from all the inputs that are coming from proprioceptors, things like muscle spindle fibers, Golgi tendon organs, some Pacinian corpuscles. Their cell bodies are out here in the posterior root ganglia. Their pseudounipolar axon brings information into the spinal cord just like any other sensory activity, but instead of ascending right away or synapsing in the dorsal or posterior horn here, these that are destined for the cerebellum actually come down and synapse with a cluster of nerve cells in the central part of the spinal cord's gray matter here. It's called the nucleus dorsalis of Clark, and it's only really found from about the T1 to L2 levels of the cord. So it's going to synapse. These new cells will stay ipsilateral, peel out over here, and ascend in the dorsal spinocerebellar tract. Now the important thing to remember about this is even though this nucleus dorsalis of Clark is spread out over the T1 to L2 area, all the inputs to it are generally coming from the lower limb, a little bit of the lower torso. And so those proprioceptors of the lower limb are what's in this dorsal spinocerebellar tract. It's located just a bit lateral to the lateral cortical spinal tract right here. But what about the upper limb? In the upper limb, we actually have something different, but kind of sort of similar going on. We have these same proprioceptors from the upper limb peel off and ascend in the cuneate fasciculus. And this is called the cuneo-cerebellar tract. It does not synapse in the cord. It actually waits until it gets to the medulla to do that. So here, let's fill in a little bit more of the brainstem. Now it's labeled over on this side. I'm going to follow it on this same side that I have here, but you can look to both sides to get a sense of what's happening. The dorsal spinocerebellar tract here in pink ascends through the brainstem, gets to the inferior cerebellar peduncle, and will then go into the fovea, the gray matter on the cerebellum, on the ipsilateral side. So that dorsal spinocerebellar tract never decussates. Likewise, here we've got the cuneate nucleus, which is going to be just beside the external cuneate nucleus. The external cuneate nucleus is where these cuneo-cerebellar tract axons are going to synapse, boom, and then they are going to head up alongside those dorsal spinocerebellar tract axons and enter the ipsilateral fovea of the cerebellum. So again, it stays ipsilateral the whole time. Now, we got another tract that we're going to talk about in detail, but it's coming from a slightly different place. We have all kinds of sensory input coming from the periphery. We're showing it on the other side, but remember this is happening bilaterally. And we have interneurons throughout the cord, not necessarily here in the nucleus dorsalis of Clark, but interneurons in this area. And the cerebellum gets information from those interneurons by means of the ventral spinocerebellar tract. And these interneurons will send some fibers, some axons, that decussate through the anterior white commissure and head superiorly in the ventrospinocerebellar tract, right here on the ventral side. And that's going to be input from interneurons, which gives the cerebellum a sense of what's happening in terms of motor activity at all times. So that's happening here. Note, it decussates as it comes into the cord, just after it synapses there. And here it's in purple. It's going to ascend through the ventrospinocerebellar tract, and it goes past the inferior cerebellar peduncle, past the middle one, all the way up to the superior cerebellar peduncle, at which point it enters the cerebellum, decussates again to get to the fovea there. And because of that, the ventral spinocerebellar tract decussates twice. Now, there is a fourth tract that I've listed on the other slide. It's called the rostrospinocerebellar tract. You can tell how important this textbook thought that was by not even including it on this figure. Basically, this tract is fairly controversial. It's also originating from interneurons in the immediate zone of the spinal cord, and it's doing the same sort of thing that the ventrospinocerebellar tract is doing for upper limbs. So ventrospinocerebellar tract, lower limb interneuron input. The rostral spinocerebellar tract, same sort of thing for upper limb interneurons. And basically, it's going to ascend near or within the ipsilateral dorsal spinocerebellar tract and then go through the cerebellum's both inferior and superior cerebellar peduncles. If this is sounding kind of nightmarish, I recommend you don't worry too much about it. It's really the least well characterized of these. And so it's either decussating twice or not at all, which means lesions affecting the cerebellum and these tracts will often be ipsilateral in nature, how they manifest. Alright, so here's the same stuff I just said. Follow it through at your own convenience if you like. So, we've talked about mossy fibers. Now let's talk about those inputs from the inferior olivary nucleus, or inferior olive of the medulla. You can see it right here, just lateral to the medullary pyramids. It's going to be receiving inputs from the cerebellum and giving outputs to the cerebellum. And it's very complex, but the take-home message for right now is it provides kind of a feedback loop for the cerebellum, which allows it to modulate motor activity. It's getting inputs from the contralateral cerebellum, and then its own climbing fibers that are coming from the neurons that are in the inferior olive will then decussate across the midline and go to the same kind of origin site, so the contralateral cerebellum, through the restiform body of the inferior cerebellar peduncle. So it's just basically going up the inferior cerebellar peduncle, and those are the climbing fibers, and they're called that because they're going to climb along the axons of some nerves that we're going to see shortly called Purkinje fibers. So now we've got an idea about how the mossy and climbing fibers get to the cerebellum, and we can kind of get a kind of fertile ground now for seeing what they do. So let's talk about the cerebellar cortex, or the fovea. The outer surface is basically a collection of gray matter, and it's got three distinct areas. And from the outside in, we have the molecular, the Purkinje, and the granule cell layers. So MPG is going to be the breakdown for that set of layers. And then we have white matter tracts deep to that. The granule cell layer down here is packed full of granule cells. They're also a type of neuron, as well as some other Golgi cells. So we've got Golgi and granule cells here. So G and G, granule and Golgi in the same area. Then out here, we've got the molecular layer. That's going to contain axons coming from those granule cells, as well as stellate cells and basket cells. And in between, we've got this really unique layer, kind of in single file, these huge Purkinje cell bodies. These are neurons. And these Purkinje cell bodies have tons of dendrites sticking up here into the molecular layer, and a single axon stretching down through the granule cell layer to the white matter. So let's take a look at the circuitry here and refer back to this illustration as we go. So granule cells, these little red guys in the illustration, are going to project their axons up to the molecular layer, but those axons then do a really weird thing. They have a little T-split and form these parallel fibers in the molecular layer. So these granule cells have an axon that splits and forms this kind of array out in the molecular layer, and this array of axons is going to stimulate the cells in this area, like the stellate cells, the basket cells, right out here, as well as the Purkinje cells, which have their dendrites up in here, are getting stimulated by it. So the stellate cells here in the molecular layer and the nearby basket cells are getting excited by these parallel fibers. And so they send their axons back to the Purkinje cells that were also excited by the granule cells. And basically, the stellate cells will synapse with the Purkinje cell dendrites. The basket cells will synapse with the cell body, or maybe even axonal synapse there. But essentially, they're going to inhibit the Purkinje cells. So the Purkinje cells had a quick on from the granule cells, and then an off that follows shortly after due to the outer and inner stellate cells, as well as the basket cells. So again, we have a quick stimulation followed by inhibition, resulting from these granule cells stimulating the Purkinje, stimulating the the stellate cells and the basket cells to then inhibit those same Purkinje cells. So quick on and off. Now nearby in the granule cell layer, we have some Golgi cells. their dendrites are going up into the molecular layer also, and they get excited by these granule cell parallel fibers, but then they actually synapse with the granule cells and tell them to cut it out. So I like to think of the granule cells as a chatty friend who really gets everybody revved up, and the Golgi cells are the friend who kind of tells them to knock it off, that they're talking too much. So what this does is we've already got the on-off of the Purkinje cells modulated by granule cell stimulation, stellate and basket cell inhibition. The Golgi cells are now actually allowing the granule cells to kind of go on-off as well, so that each one of their stimuli is distinct. So they're on, off, and it's clear when one stimulus happens and when another doesn't. Now, that's going to be the inputs from our granule cell. Let's talk about those mossy fibers. They're going to be shown here in red. These mossy fibers from the pons and from the spinocerebellar tracts come up, and in the granule cell layer, they make what's called a cerebellar glomerulus. Kind of its big extended axon here is a big blob that we call the cerebellar glomerulus, and it's going to have excitatory synapses with the granule cells. And so, basically, they're going to be the things that cause these granule cells to fire in the first place. But this is the same basic area where these Golgi cells are telling the granule cells to knock it off. So again, stimulation, but inhibition tied very tightly to it. Over here, we've got the climbing fibers coming from the inferior olivary nucleus. These are called climbing fibers because they kind of climb or kind of roll along the Purkinje cell axon, and they're heading up into the molecular layer, and they're going to strongly excite the cells that are there. So the climbing fibers are heading up to the deep cerebellar nuclei, bam, just like the mossy fibers. Then they go up to the cortex here, bam, and they're going to excite the Purkinje cells. Now, the Purkinje cells, right here, these big blue guys in the middle, those are the only outputs from the cortex. Every other cell in this area is modulating the output of the Purkinje cells, and the Purkinje cells are going to send inhibitory synapses, or pardon me, inhibitory signals to synapse with the deep cerebellar nuclei. So let's talk about those. We've seen this view already, but to see the deep cerebellar nuclei, no big surprise, we have to actually cut the cerebellum open and look on the inside. So here we have a stain through this area right here showing the deep cerebellar nuclei. They're these structures here. Now you can see the squiggly dentate nucleus, but I'll forgive you if you can't quite make out the others. Those are going to be called the vestigial, globose, umbiliform, and dentate, the one we can see out here. So going from lateral to medial, dentate, umbiliform, globose, vestigial. Don't eat greasy foods. Best mnemonic I've heard, can't really improve on that. So let's take a look and see what these look like in a clear stain. We have the dentate, the umbiliform, the globose, and the vestigial. Now they don't look like much aside from the dentate, but functionally they're very important. These are going to be excited by inputs from mossy and climbing fibers. They're then going to be inhibited by outputs from the Purkinje cells coming from the cerebellar folia there. And then the deep cerebellar nuclei will send their axons out of the cerebellum, and they are the only outputs from the cerebellum as a whole. Now, real quick, you should know that the globose and umbiliform are very functionally similar, and they're also sometimes referred to as the interposed nuclei, which we'll be using hereafter. So, the dentate nuclei, the biggest one, is going to receive its inputs, those inhibitory signals from Purkinje cells, from the lateral part of the cerebellar hemisphere. So right here, this region of the cerebellum projects to dentate nuclei, and it's going to be most involved before movement even happens. So the premotor organization of motion. So tied with our premotor planning area, the frontal lobe, to initiate and plan motor activity. And in that, it's a little bit tied to the function of the basal ganglia, which we'll talk about at the latter part of this talk. The interposed nuclei, that's the umbiliform and globose put together, receive their Purkinje cell inhibitory signals from the intermediate part of the the intermediate part of the cerebellar hemisphere. So right here, this is the part projecting to those interposed nuclei. This circuit is involved mostly when we have ongoing motor activity of the limbs, and it's going to help us make those motions of the limbs, the upper limb and lower limb, smooth and coordinated. Likewise, the vestigial nucleus is getting its inputs from part of our vermis here, as well as some of that flocculonodular lobe we talked about earlier. And it's going to be involved in coordinating ongoing movement and motor activity, making it smooth, involving the trunk and core muscles. So very core muscles are going to be getting their modifications and their edits from the flocculonodular lobe by way of the vestigial nuclei, while the upper and lower limbs get their edits from the intermediate part of the cerebellar hemisphere by means of the umbiliform globose nuclei, which together are known as the interposed nuclei. So now let's talk about how the dentate nucleus is actually able to do what it does. So on this slide, we've got the lateral cerebellar hemispheres and dentate nucleus represented here. So lateral cerebellar hemispheres project Purkinje cell inhibitory axons to the dentate nucleus. Now the dentate nucleus wants to help us plan and coordinate new motor activity and really smooth it out. So to do that, we're going to have the dentate axons travel to the ipsilateral, same side, superior cerebellar peduncle to get to the midbrain, and at that point it's going to decussate, cross the midline, and get to the contralateral thalamus. Now in the thalamus we have VA, but particularly ventrolateral, VL, thalamic nucleus there, and it's going to synapse with that nucleus, and those new axons will then go up to the same sides, premotor planning area as well as the primary motor cortex, so that we're getting a sense of how to plan a motor activity that does what we want it to do. And that's largely what the dentate nucleus is all about. Thereafter, we're going to have the motor planning trigger strongly the upper motor neurons in the pre-central gyrus, primary motor cortex, which will go through the corticospinal tracts to get to the spinal cord and actually make motor activity happen. Now, not represented on this picture is a second target. There are, after we have the decussation in the midbrain, so we're going to have these axons go through the superior cerebellar peduncle, decussate. Another target for them, aside from the thalamus, is the contralateral red nucleus. The small cells in there, parvocellular, small cell, part of the red nucleus, those are then going to project inferiorly to the inferior olive. And those are the inputs to the inferior olive that are then going to be processed. Then we're going to have those olivary climbing fibers cross the midline. They're going to decussate to the contralateral cerebellum and basically wind up on the same side where this initial depolarization or circuit happened or initiated. And that's basically where we think motor memory is happening. So when we do motions that are very complicated and we don't have time to think through every small little motion we have to make, but we have a coordinated, practiced motion, it's probably very much due to this activity of the dentate nucleus, lateral cerebellar hemispheres, the red nucleus, as well as the inferior olive, getting us to coordinate those motions so we don't have to think them through. We can just do them through consistent practice. So deep breath. That's it for the dentate nucleus. Now we're going to move on, same basic idea, to the interposed nuclei. So from the intermediate cerebellar hemisphere, we're going to have Purkinje cells projecting to the interposed nuclei, and we'll put vestigial in there too. We'll see it on the next slide in particular. And we have basically the same starting point. We have those axons traveling from the deep cerebellar nuclei, the interposed nuclei, through the superior cerebellar peduncle, decussating in the midbrain to reach the contralateral thalamus. And it's very similar to what we just saw. It's going to synapse with neurons in the VL, ventrolateral thalamic nuclei, which will then send their axons to the primary motor cortex. Now, we're simplifying this a bit, but I think it works out, because what we're trying to do here is modulate and change existing motor activity. So to do that, we have to compare those inputs we got from the pons about intended motor activity to what's actually happening, and then make some changes. And those changes are going to be going to the primary motor cortex. That's going to go through the corticospinal tracts, largely lateral corticospinal tract, to the cord, The spinal cord then is going to make motor activity of the muscle happen, but it's also receiving sensory inputs from the muscle, and some of those are proprioceptive inputs that are traveling through the spino-cerebellar tracts, and that's how the spinal cord is aware of what's happening and is able to compare intent from the pons by way of the primary motor cortex to actuality, and then follow this circuit to actually affect change in what the spinal cord is telling the muscles to do, and that's how coordinated motor activity happens. Similar to before, we have another target as well. We have these same axons from the interposed nuclei going up the superior cerebellar peduncle and crossing over in the midbrain to get to the contralateral red nucleus again, but then they're going to go to the bigger cells there, the magnocellular part of the red nucleus, which is going to give rise to the rubrospinal tract, which decussates and descends in the spinal cord and helps innervate some flexors of the upper limb. Not a major function for us humans, but it is still happening. All right, moving on to the vestigial nucleus, the last of our deep cerebellar nuclei. So here, the vestigial axons have a slightly different way of getting things done. They're going to coordinate motor activity of the trunk. So here we have the vestigial axons getting their output from the vermis and floccular nodular lobe Purkinje cells, projecting to and inhibiting the vestigial nuclei, and the vestigial nuclei are then going to travel through the ipsilateral superior cerebellar peduncle and decussate in the midbrain, just like before. So same initial step as the last two, going to the thalamus, and because it's doing the same thing as the last slide, only we're affecting the trunk instead of the upper limb and lower limb, we have a very similar circuit. It goes to the thalamus, VL thalamus nuclei, go to the primary motor cortex, and make changes happen to trunk muscles by affecting signaling of the spinal cord to those muscles. Now, here's the other thing though. Vestigial nuclei do not only project up to the thalamus, they will also go through the inferior cerebellar peduncle, kind of in reverse. They'll exit through the inferior cerebellar peduncle, which is specifically known as the juxtarestiform body, those fibers going the opposite way. So juxtarestiform body is going from the cerebellum out to the medulla, and it's going to reach the ipsilateral vestibular nuclei. So those vestibular nuclei in the medulla are receiving inputs from there. Vestibular nuclei are then able to suggest changes to the spinal cord's activity through the vestibulospinal tracts. And so that's happening. But one thing I want to draw attention to is that these outputs to the vestibular nuclei going through the juxtarestiform body are ipsilateral, but we have the same nuclei going through a little structure called the uncinate fasciculus to reach the other side as well. So we basically have bilateral projections of the vestigial nucleus to the right and left vestibular nuclei, which then suggest changes to the spinal cord on both sides. So it's keeping the body upright. So we have bilateral inputs. Vestibulospinal tracts hit the spinal cord, and they again suggest changes to the muscle, and that's how we're going to wind up having our balance affected by both our cerebellum to make things smoother, by means of the vestibulospinal tracts to actually make muscles fire and actually do something. Now, point of fact, there are some vestibular nuclear inputs to the cerebellum as well. We'll just note that they're there to let the cerebellum know what's going on, but leave it at that. You thought we were done because we ran out of deep cerebellar nuclei, but I have some bad news. We got one more, because some of these axons from the floccular nodular lobe and the inferior part of the vermis don't even bother synapsing with a deep cerebellar nucleus. They just go to the vestibular nuclei and treat them effectively as deep cerebellar nuclei, except they're not in the cerebellum, they're in the medulla. So what they do is they're going to bypass the vestigial nuclei entirely, travel through the inferior cerebellar peduncle outwards, that's that juxtarestiform body again, and get to the vestibular nuclei. Now this is happening bilaterally, and really don't worry about how. Sometimes it uses the other vestigial nucleus as a relay point to get to both sides, but essentially from the vestibular nuclei we're going to have outputs to a variety of places. Two of them are things we've already seen. The vestibulospinal tracts, the medial vestibulospinal helps us move our head and neck in response to how our eyes are moving to keep things aligned so we can keep something in view. The lateral vestibulospinal tract coordinates primarily extensor movements of the lower limb to keep us balanced. And we also have the medial longitudinal fasciculus stretching up from the vestibular nuclei to cranial nerve 6, 4, and 3, so that movement of our head is taken into account as we coordinate movements of our eyes. So that's basically what's happening with this last pathway from the cerebellum out to the cord to affect motor changes either in the muscles of the neck and head for the medial vestibulospinal tract, or the lower limbs primarily for the lateral vestibulospinal tract. And as before, there's going to be some vestibular feedback to the cerebellum to let it know how it's going. Alright, so we've worked our way through all that misery, all that pathways, all that circuitry. Let's talk about what happens when the cerebellum goes wrong. When we have dysfunction of these spinocerebellar tracts, or the peduncles, or the cerebellar kind of structure itself, the folia or deep cerebellar nuclei of the actual cerebellum. Basically, it's going to cause motor coordination problems, and it's generally going to be on the ipsilateral side, because inputs from to the cerebellum tend to be ipsilateral only, or to decussate twice. So either they don't decussate at all, or they decussate 2 times. So damage to the cerebellum tends to cause problems with coordination on the ipsilateral side. Now, if we have damage to the midline vermis, the very close midline trunk structures that are going to project to both vestibular nuclei, then we're going to have bilateral ataxia, or uncoordinated motion of the limbs, particularly the trunk muscles on both sides. And that is what ataxia is. It's an uncoordinated movement caused by problems with the cerebellum. It can affect the trunk, and you can see here, it makes sense that the trunk would be primarily affected by things coming from the vermis, or the flocculonodular lobe, the deep cerebellar nuclei, the vestibular nuclei, things like that. And basically it's going to make balance very difficult. To test this, somebody might be asked to walk heel to toe in a straight line. Balance is very difficult even if you have a functional cerebellum. Or to stand with their feet close together, narrow base, and eyes closed. This is known as a Romberg test. And people tend to fall towards the affected side. Now, if we have damage to the lateral part of the cerebellar hemispheres, or some of the spinocerebellar tracts coming from the limbs, and then particularly the dentate deep cerebellar nucleus, we might wind up with appendicular ataxia, or ataxia of our upper limb and lower limb. So that's going to be uncoordinated movements of our extremities. Ataxia is a biggie for cerebellar dysfunction. Let's bring up a few others. Dysmetria, overshooting or undershooting when you're trying to touch moving or stationary objects. So taking your finger from your nose and touching the finger of someone else, and they keep on moving the finger. If you can't quite do it and you keep under or overshooting, that's dysmetria. Dyssynergia is the breakdown of a complex movement into individual parts. So if I'm trying to reach out and grab something, if I have to make 3 steps to finally grab that thing, that's dyssynergia. I'm not synergizing those movements into one smooth motion. Dysrhythmia, weird timing or pacing of movements, and my personal favorite, dysdiadochokinesia. This is a pretty clear sign of a lateral cerebellar lesion because someone's unable to make rapid alternating movements. So you have a person put their palm down on their thigh on one side, palm up on the other side, and just flip their palms up and down, rapidly pronating and supinating. I'm doing it now, maybe you can hear. And if you can't do that, that is dysdiadochokinesia. Now you may notice when I described ataxia and dysmetria, that's the same sort of thing that people do during a roadside sobriety checkpoint, and that's because the cerebellum is one of the first things affected by alcohol. So intoxicated people actually tend to have what appears to be cerebellar dysfunction. Now, before we finish, let's go back to where we started and make sure that we're covering the blood supply here as well. So we can see that the superior cerebellar artery is covering this superior part, going to be both the superior vermis, intermediate and lateral part of the hemisphere. Then we've got the posterior inferior cerebellar artery taking care of a large portion of the more inferior vermis, as well as the intermediate and lateral parts of the hemisphere here. And the ICA, anterior inferior cerebellar, handles some more of these midline internal structures, but otherwise it's not really going to be taking up as much space as the others. But you can see that it does cover a lot of those cerebellar peduncles. That can be very bad if you have a lesion involving the ICA, and that's going to cause problems with the inputs and outputs of the cerebellum. Alright, so now let's talk about the basal ganglia. Essentially, the basal ganglia are a bunch of nerve cells inside the cortex. They're not technically ganglia since they are within the central nervous system, but that's the name we've stuck them with. They are the caudate nucleus, which is a large, basically tadpole-shaped gray matter collection of cell bodies that is in the wall of the lateral ventricles. It gets very large anteriorly in contact with the putamen and the nucleus accumbens, but as you go further posteriorly along the lateral ventricles, it gets smaller and smaller as you move from its body to its tail, and it gets very small but still remains even in the temporal lobes as a small bit of gray matter in the lateral ventricles. Now the putamen is actually linked by the little bridges of gray matter with the caudate nucleus, and that makes sense because they actually function very similarly, and together they're referred to as the striatum. So even though it's a distinctive structure visually, it actually functions along with the caudate nucleus. Now the globus pallidus is a functionally distinct structure that's located medial to the putamen. If you're very lucky on a section, you might see both the external and internal segment, or the pars externa and pars interna. And because they're located close together, you'll sometimes hear the putamen and globus pallidus together referred to as the lenticular or lens-shaped nucleus. Now going into the midbrain, we've got the substantia nigra. It's functionally part of the basal ganglia and has two parts, pars reticularis and the pars compacta. And then, inferior to the thalamus, but superior to the substantia nigra, we have the subthalamic nucleus. So all of these structures have interconnections that allow us to plan and eventually initiate motion. On this slide, what I'd really like you to pick up is that we've got inputs to the basal ganglia, mostly to the putamen and the caudate nucleus here. They're going to be the major receivers of inputs, and then they're going to parcel that through the other nuclei, and the major outputs are going to be GABAergic. So they're using an inhibitory neuron that's going to the VA and VL thalamus. And so those outputs are going to be coming from the internal segment of the globus pallidus and the pars reticularis, which again they're not showing here, which is a bit unfortunate, but the pars reticularis of the substantia nigra. So those GABAergic outputs are going to go to the VA and VL thalamus. And the VA and VL thalamus are largely involved in stimulating motor activity in the frontal lobe. So generally, VA and VL thalamus stimulate motor activity. The basal ganglia, their tendency is to inhibit motor activity by inhibiting the VA, ventral anterior, and VL, ventrolateral thalamus. Now, that's the quick breeze through this. We're now going to go through the two major pathways through the basal ganglia called the direct pathway and the indirect pathway. The absolute most important thing to remember in this is that the direct pathway, activity through it tends to increase motor activity, while the indirect pathway tends to decrease motor activity. Everybody good? All right, let's go for it. So the direct pathway is shown here on the left. And let's just follow that through. So we've got cortical neurons extending to the striatum, and they're being stimulated with glutamate. And so that's stimulating the striatum, aka the putamen and globus pallidus. These striatal neurons are going to send inhibitory signals right here. These inhibitory signals are then going to be going to the globus pallidus, pars interna, and the substantia nigra reticular area. That's using GABA and substance P. These are inhibitory neurons. Then the globus pallidus, pars interna, and the substantia nigra, pars reticularis, are going to send inhibitory GABAergic signals to the VA and VL thalamus, which, remember, stimulate motor activity. So the long story short is that by inhibiting the inhibitor of the VA and VL thalamus, the direct pathway actually increases motor activity. Now, on top of that, we've got dopamine-releasing neurons from the pars compacta of the substantia nigra. So SNC right here, those dopaminergic neurons are going to bind to receptors in the striatum, and they are actually going to increase, that dopamine increases activity through the direct pathway. So substantia nigra will actually make the direct pathway work better by increasing the inhibition of the inhibitor of the VA and VL thalamus. So long story short, as before, Direct pathway makes motor activity more pronounced, and substantia nigra's dopamine makes that happen even better. Good? All right. Let's scoot on over to the indirect pathway, shown here on the right. So we start off the same way. We've got glutamate from the cortex going to the striatum. Now here, signaling through the indirect pathway is kind of interesting in that we start the same way. We've got GABA going to a slightly different target along with another new neurotransmitter called enkephalin. Now right here, that is not going to the same target. It's instead going to the globus pallidus external part, which is then, it's inhibiting that, which normally inhibits via GABA the subthalamic nucleus. Then the subthalamic nucleus generally is going to activate through glutamate the globus pallidus pars interna and the substantia nigra pars reticularis. So right here we're at the exact same part, but now what we've done is we've inhibited neurons that inhibit neurons that excite neurons that inhibit the VA and VL thalamus. Now, if you are mad at me for saying that last sentence, I feel your pain, but we're basically stuck there. The long story short is indirect pathway activity will tend to decrease the amount of activity from the VA and VL thalamus, which will then result in a net decrease of motor activity. Now, dopamine plays a role here as well. So the same substantia nigra pars compacta neurons that release dopamine are also affecting the indirect pathway, but they affect it through different receptors, which actually inhibit the striatum and make the motor activity a little more plausible. So once again, dopamine is there to help increase motor activity from happening because dopamine will largely inhibit the indirect pathway. Review that last slide until it makes sense or until you just can't take it anymore. Because here's the payoff. Parkinson's disease. In Parkinson's, we have the death of dopamine-releasing cells in the pars compacta of the substantia nigra. So we lose these cells. And what happens? Well, we stop activating the direct pathway as heavily as we did. We stop inhibiting the indirect pathway as much as we did. And we're going to wind up, in essence, causing a net increase in inhibition of the thalamic nuclei and the cortical motor areas. So with Parkinson's, we've basically decreased the activity of the direct pathway and increased the activity of the indirect pathway. So people with Parkinson's are going to tend to have a lack or a paucity of motion. They're going to move with small shuffling steps and kind of a gait that's very, very, very ponderous, little tiny steps. They tend to have very masked and unreadable faces because their facial movements are just not spontaneous anymore, and they don't generate spontaneous emotional expressions. So they can make willful motions, but it takes a lot of effort, and it's very constrained and minimal when it does actually happen. Now, when these people actually relax, they tend to have a resting tremor affecting the hands, upper limbs, lower limbs of the mouth. That's not related to an increase in motor activity. It's just an inhibition removal from the lower motor neurons of the spinal cord. Now, one thing to note is that the basal ganglia, and especially the substantia nigra in this case, tend to have effects on the opposite side since they're inhibiting the output to their contralateral cortex, which will then decussate when we go from the cortex down to the lateral cortical spinal tract. So just be aware that Parkinson's disease, if it's related to one side, loss of the substantia nigra will affect the contralateral side. All right, if you can handle that, you can handle this. Huntington's chorea. So chorea means dance or writhing motion, and this disease is due to mutation, a trinucleotide repeat that's going to result in the death of the neurons in the caudate nucleus and the putamen. So these striatal neurons are going to die, and the enkephalinergic inhibitory neurons that are here, releasing enkephalin, are particularly affected. The net result of this is basically the opposite of Parkinson's. You're going to wind up having increased activity of the direct pathway and decreased activity of the indirect pathway. And that basically means that you're going to wind up with increased and uncoordinated, unplanned, unintended motor activity. So limbs are just going to start writhing around. This is often accompanied by personality changes because the basal ganglia do more than just motion, although that's what we're focusing on right now. As before, since the basal ganglia will project to the ipsilateral cortex, but the cortex will then decussate on its way to the spinal cord, you tend to have unilateral issues with the basal ganglia and the striatum here manifest on the opposite side if there's a difference in the way that they're dying off. Now, one thing about this that's interesting is the caudate nucleus is pretty huge, and it's in the wall of the lateral ventricle, and as those cells die, you can actually see the ventricles enlarge. So ventriculomegaly or hydrocephalus ex vacuo is what that's referred to. It's not due to obstructive blockage of the drainage of the CSF from the ventricles, so it's not obstructive hydrocephaly. It's only appearing that way because the lateral ventricles are getting larger because a big structure in their lateral wall is dying. All right, last we have hemibolismus. So hemibolismus is very much like Parkinson's in that it's an increase of motor activity. However, it's very different in that a different structure is affected. It's going to be the subthalamic nucleus, and it's particularly characterized by flinging of the proximal segments of the limbs. So if someone kind of puts their arms in a chicken dance pose, so elbows flexed, hands in front of shoulders, they'll start kind of waving their arms uncontrollably. So the subthalamic nucleus, which typically is going to stimulate the internal segment of the globus pallidus and pars reticularis of the substantia nigra to inhibit motor activity, removal of that is going to wind up increasing motor activity through the indirect pathway. So that's basically an increase in motor activity because of a different structure than we saw in Huntington's. And as before, since it's going to be affecting the basal ganglia on one side, if it's a lesion that's affecting one side or death of the cells there due to a vascular infarct, you're going to wind up with contralateral signs as it goes to the thalamus, goes to the cortex, but then the cortex decussates on its way to the spine. All right, it's been fun. I hope this topic makes at least a little more sense than it did before, and I'll see you guys in one of the other videos.